Right, I'd now like to invite Dr. Thomas Thompson from Queen's University, Belfast.
I want to start by thanking organisers for putting up with my very busy schedule. I was meant to be tomorrow, but thankfully they squeezed me in. So I can talk to you today about some of the work we've been doing here in Belfast, and that's primarily using the idea of cold plasma to both break down biofilms and also increase susceptibility of things like antibiotics. So as we study biofilms, our idea, our definition continues to expand. We've gone a long way from these big mushroom-shaped, stilly sort of base models of biofilms to these more aggregate type cells. And as Dell rightly pointed out, the mechanisms at which biofilms survive are largely tolerance mediated, things like efflux pumps, matrix, persister cells. These aren't genetic components, they are tolerants. And that's important because unlike resistance, tolerance is reversible. And in the work in our lab is we aim to use things like cold plasma to break apart some of these really nasty infections and see if we break apart biofilms, can we use typical clinically used antibiotics and can we make them work again? So if anyone's not familiar, this is what cold plasma looks like. This may or may not play. I don't think they're going to play. Okay, but essentially, okay. No. Oh, there we go. Thank you so much. So this is like a plasma bullet here, and literally, while it looks like it's a constant stream, it's literally loads of little plasma bullets. They bombard the surface of biofilms, but the plasma comes in lots of different forms. We have these that look like little lightning bolts here, and these DVDs, these dielectric Bayer discharges, and we use these, and to bombard the surface of biofilms. It's called plasma, not like blood plasma, because it's the fourth state of matter, and effectively, if you take a gas and continue to heat it, you get this fourth state, which is a mixture of reactive oxygen, nitrogen species, and it's those reactive species which are essentially doing the damage to biofilms. And so what we want to know is we want to know how biofilms are recovering from plasma. So we did some RNA-seq. Actually, what you see at 1 hour, 4 hours, and 24 hours, you get this nice shift where 1 hour post-plasma treatment, there's a huge downregulation in genes related to things like oxidative stress, which you expect. By about 4 hours, we see as upregulation, and by 24 hours, things are kind of back to what we call homeostasis or oxidative hemodynamics. And so we want to know, well, could you monopolize in this? And so we found a few nice key features. So at 1 hour, we see a huge downregulation in genes related to ribosomes. So the ribosomes are being shut down, and that's one of the hallmarks of these cells are about to go into persister formation. We also saw a huge shutdown in genes related to iron, and any upregulation was to do with iron storage. So these cells were simply, we're going to get rid of any free iron, because if you hit iron 2+ with any oxidation, you're going to get iron 3+ plus extra reactive species. So it's to stop that secondary reaction, we call it the Fenton reaction. At 4 hours, we saw really interesting things, and this is touched about today better. We saw this huge upregulation in 12 to 14 genes, all upregulated with prophage. At these ways, they were not entirely clear, but what we do think is happening is either that these phages are either jumping ship when things get bad in staph aureus, but that might have the added consequences where you get this lytic cycle of phages causing additional damage to biofilms indirectly, and something we weren't really expecting. And again, this was kind of shown previously in an E. coli paper as well. We also see changes in staphyloperidin, so a syrophore. So we actually see downregulation in staphyloperidin A, which doesn't work well in anaerobic conditions, and we see upregulation 4 hours later in staphyloperidin B. It actually makes sense because plasma generates this sort of acidic anaerobic environment, so the cells are wise enough to change the different type of iron to scavenge it away again. And also then we see this huge downregulation as well of virulence genes and ARG signaling. And so what this really means is if you use this understanding, you can sort of predict essentially what drugs will work well against plasma and biofilms. So if this is our typical kill curve, you get this sort of typical biphasic kill. And if we treat these biofilms for about 2 minutes, so we take this 120-second time point, which is equivalent to about 2 log reduction, and then we treat it with something like ciprofloxacin, you get this huge reduction in the amount of ciprofloxacin you need to eradicate this biofilm. At the minute, we're putting this down to the fact that you have increased access to the cell because the cells are becoming leaky as you blast them with plasma, but also there's changes in sort of detox genes as well. And we saw something similar with tetracycline. Again, we're putting this down again. increase access to sort of the sites like 30S ribosomes, but also the ribosomes are a bit more leaky now. And again, this is overall amplifying the effect of tetracycline. It's not universal, and we were kind of glad to see it because it doesn't work in things like amoxicillin. And at the minute, that kind of makes a bit of sense because amoxicillin works better on growing cells. In this stage, this biofilm has, after being hit by plasma, started to really hunker down and form sister cells. There's less growing cells, there's less peptidoglycan membranes for amoxicillin. They work, so you wouldn't expect to see synergy. But really, putting this all together, our response is a sort of it fast and then it fades. You get huge shutdown at 1 to 4 hours, and then things look normal by 24 hours. And I guess the importance of that is if you don't look at every single step, if you just blasted a biofilm and then look 24 hours later, you would see no change. And using things like omics is a really useful way to predict potential synergy, and that's what we saw here. We saw there's better access and lower defenses, and potentially, yes, amoxicillin doesn't work in this early window, but by changing that, maybe it would work later on. And overall, we can really use, and there's a bunch more on this as well, cold plasma really is a really useful way to actually remove some of those biofilm tolerance mechanisms and actually start to make antibiotics really effective again. And so I want to thank all people Queens and our collaborators at UCD and Thomas Jefferson. Thank you.